Welcome everybody to the session. So today's session we'll be covering a cloud-enabled generative AI framework for protein homology and antibody neutralization prediction. My name is Shivam and I am currently a software engineer at AWS in Seattle, USA. To begin with, the strategic objective of this presentation is for of this project was for transforming proteomics through Gen AI and cloud computing to predict protein homology and antibody neutralization with near experimental accuracy. Uh, to go into the meanings of these words, first of all, Gen AI. Gen AI are systems that have the ability to create new content. For this project, Gen AI was used to generate the data and the essential steps within the procedure for prediction of protein homology. What is protein homology? So basically, whenever in the industry anyone is making antivirus, for example, we had COVID, we had to make an antidote for it, we had to make a vaccine for it. Whenever we are making a vaccine, we each and every step in that vaccine making goes through a series of steps that is basically mapping out what would be the ideal chemistry of the antibodies that need to be put in that help in making that vaccine. So this project helps us to speed up that process so that in order we can make vaccines faster and save lives. The core innovation of this project relies in the integration of protein language models, PLMs, and structural diffusion to bypass traditional computational bottlenecks. Basically, the steps that were earlier a barrier uh, uh, can be overcome and we can speed up the process. So what the core problem is, the scaling problems. The traditional systems that we earlier had require weeks of compute time for a single complex. And if in the world we had another emergency like the COVID-19 that happened, we need speed is of the essence. We need to come up with a vaccine fast. So this technology can help us cover that gap and basically reduce that time frame of, let's say, 3 months to 1 week or a few weeks, and speed up and get us those accurate results that can speed up that vaccine. Again, this doesn't replace the essential trial process which goes into making of a vaccine. It just help us come to the right set of chemicals that would help us in making the antibody faster. So it doesn't bypass any of the steps that are necessary for human safety. This only helps us in coming up with a solution faster. This has been a process like of protein comparison for decades now. Since vaccines have been formed, they had to come up with an effective pipeline in order to come up with an effective way to come up with vaccines faster. So in the 1990s, they used to use a BLAST algorithm, that is sequencing identity by local alignment strings. Basically, one-to-one mapping of each of those proteins and DNA sequences. Then in 2010s, we came to a more statistical method with the evolution of like computer system and data science, which sped up the process. But with the today's computational accuracies and the computational power that we have with large language models, we can feed those input system with a lot more of the data that we already have, and it can help us in coming up with a solution much faster, because all the evolution that has gone on till now in this field, basically in the chemistry of those vaccines, can be utilized and can help us come up with an antidote or the vaccine faster that can help tackle that particular disease. This is COVID-19 is just a bigger example, because that was one of the recent viruses that required us prompt action. But even for the other things that we are diseases that we are making vaccines for. Years and years of research goes into them. This is just a small step to reduce the amount of time that helps us reach that effective vaccine. So how does this generative AI architecture works? First of all, transformers, which is a Gen AI term that is basically learning the grammar of life through self-attention and sequence data. Basically, the data that is fed into the learning system that would help us predict it. And then diffusion is generative 3D coordinates. So end of the day, all of these Gen AI models that give us uh, the results are dependent upon some kind of a mathematical formula. That is, and when we realize this 3D dimension, that is 3D dimension to the points. Each point is given a different weight for those antibody structures, and that help us. The more the dimensions are, the more accurate the result becomes. 3D for this study was used because that is the largest data set that we have. As we go into smaller niches, this model is still effective. It's just that we wanted to through our research show one of the biggest impact, that one of the biggest data sets that is available with the most amount of data can be used. And graph neural networks is basically a feeding methodology with Gen AI agents. All the this process becomes automated. What it means is, whenever a new kind of a sequence is comes up in the market or through various research studies that is continuously carried out by various academic institution, our agents automatically pick that up and feed it to the neural network. The neural network gets trained on it, and any future drug that needs to be produced relies on the new data, and human intervention is reduced. Just let's move to the deep dive of what this protein homology is. So basically, functional homology detection. Our framework embeds protein sequences into manifold where proximity correlates with function rather than just the sequence identity. This allows for detection of structural mimics across different species that traditional sequence alignments would miss. What that means is, whenever there is a disease, it has some kind of a similarities to other. For example, as I've been using the example of COVID-19, COVID-19 was closely able to correlate to some of the influenzas that we have in the past. So if we use those data for those vaccines that was created for that influenza, we can come up with the solution faster by just mimicking those data points and seeing what becomes most effective. Moving on, this is what the accuracy results are. So as I had earlier shown, there have been years of evolution. The oldest one, blast sequence, had a 42% accuracy. Whenever a vaccine is being produced, it is a trial and error using various kinds of protein sequences that are out there while making the drug. And if it's a miss and we keep on missing, it takes longer time to experiment and come up with the right vaccine. End of the day, the vaccine that is given to the humans is the one that is the most effective. This is the process that comes in while making that vaccine to come up with that effective vaccines. This project aims to improve that pipeline and make it more effective. Moving to the core concepts of antibody neutralization. So, as I said, at the end of the day, all of these neural networks at the end have some kind of a math that helps us predict it. At the core fundamental level, the neutralization is a function of that equilibrium constant that our AI predicts free energy, which is basically the randomness those protein sequences have, because those protein sequences have some amount of randomness in them. Basically the difference, because each even though COVID-19, let's say, is closer to an influenza, it isn't exactly an influenza. It has correlation to it. So this mathematical function basically helps us to account for that randomness and come up with the effective vaccine solution. And this is the formula: antibody into antigen upon the complexity. Complexity is of the protein sequence that we are calculating. This is like state of the art modeling techniques that are available in the market, but have been bounded by the technology that is available. But as we come up with this Gen AI network self learns, so it takes away that barrier. Basically, the antometer extracts fine grain all atom level features, which traditional computers would take a lot of time. By separating out the data and allowing parallel processing, we can speed up that process massively. And antibody and antigen complexes using geometric graph learning and multi-scale attention. That is at a core level, again, as I said, different dimensions, correlating the bonds between each of them and finding the more effective solution. And RF2 antibody, this is Rose Tierfold fine refinement meter, which is industry standard while calculating the effectiveness of the vaccine and what are the different protein compositions that need to go into it. How does cloud come into the picture with all of this? I talked about like state of the art models that are available, and like how the entire process works. Now coming to the core concept, like how does cloud computing and today's technological innovations help us in scaling this? Basically microservices. Nvidia comes up with very good chips that can speed up the production, because end of the day, the amount of data that we had is humongous, and each time that we run a simulation, it takes days of data. Some of the most effective machine learning models that we have, if we fed it all the data that is available today, it would take about 15 to 20 years to come up with the effectively trained models. But if we parallelize the process, we can come up with much more. And with GPU acceleration, end of the day, all of these calculations that go into predicting and coming up with the solution happens on the GPU level. GPU acceleration techniques can help us speed that up. And global data lake, that is basically everything that is fed into the system has to be present in one place, because the more places and staggered it is, we cannot trust each and every source, because some malicious agent might be adding the wrong data that would end of the day reduce the effectiveness of our model and make coming up with the solution slower. That is why a global data trusted data structure would be made, which would basically go through a variety of security agents, security GenAI agents that basically have the capacity to detect malicious data, and if something wrong is being fed into the system, and would flag it. So as I said, where at the core levels, at a very small level, the traditional homology search used to take 40 to 45.2 minutes. With our AI enhanced technology, it came down to 1.8 seconds. The structure fold, which used to take 12 hours, came down to 35 seconds. Docking simulation, 72 hours, came down to 12 minutes. Neutralization prediction, 2 weeks to 4 seconds. Real time speed up. This is the benchmark and the innovation of these GPUs and these modern technologies have. If we can reduce all of these at a massive scale, this 45.2 minutes is for one small example. Across the board, it can be much larger data sets, but this is the amount of speed up that we can provide to those structures, and end of the day, come up with a solution much effective and much faster. And this is how the trends are going. If you see, this has been the trend, and this correlates with the coming up of these new Gen AI models and innovation in the chip technology that allows us to support that. The case study that was done for this was the viral defense, basically rapid response pipeline during 2025 influenza mutation outbreak. Our framework identified 3 prospective neutralizing antibodies within 48 hours of the sequence release, which is traditionally takes about 2 weeks to 48 hours to come up with a first solution, and 92% correlation with subsequent in vitro neutralization assays. That is the eventual solution that was come up with. We had a 92% accuracy within the first go, which reduced approximately 9 months of computational time, which shows us again the effectiveness of this model. 98% reduction in compute cost, because with the accelerated framework that we have and the Gen AI sequence, it also prioritizes frugality, because not every company or every academic institution has that kind of money to come up with billions or millions of dollars of data. This reduces the cost of getting that impact as well, from about $4,500 to $0.12. So 98% reduction gives those talented scientists who might be able to come up with the solution but were stranglehold basically by the cost of it, now get the increase and boost in their research funding, because what earlier cost $4,500 now just costs 12 cents. And from a strategic overlook as well, this helps us at a universal level because vaccines are universal and speeds up that entire process. Indian, I would like to quote one of the Global Biosecurity Summit quotes: "The democratization of generative protein design offers unprecedented medical hope, yet necessities of a robust new frameworks for biosecurity and AI safety." What it means is we have an unprecedented amount of growth in and potential in the ways we can come up with it, but this has to be done in a safe and effective way so that we are sure that the data that these models are being fed is not with malicious intent and it is being used for the correct purposes. Thank you so much, and I would be happy to take any